Hello student, today we start the BSc second year paper 7 fish developmental biology. Paper second, paper number 7 fish developmental biology. Okay, within that the unit number 1, that is the developmental biology, already we are completed. Then we are started, that is unit number 2, that is developmental biology. Within that, morula, blastula, fat map of blastula, already we are completed. Unit number 2, bit number 4, that is the gastrulation. Development, nothing but the development process, development program, development progress of all these things. Morula, blastula, fat map of blastula. Then the position is, that is the gastrulation. Okay. On my right hand side, if you see the figure of early stage of gastrulation under low magnification, and B figure, that is the posterior end under high magnification. Figure indicate all things. I will explain one by one. Gastrulation is a dynamic process that involves various morphogenetic movement of the prospective cell. During this stage, the cell which were arranged in earlier stage get rearranged and recognized to transfer the deposition of embryo, important thing point of view, that is deposition of embryo into the body plane of adult fish. Okay, on the right hand side you see it is the blastoderm, blastocele, investigating endoderm. This is the A figure. Lastly, the yolk, blastoderm, investing endoderm, periblast, and this one is the yolk. Figure number A and figure number B indicate that early stage of gastrulation. A under the low magnification, and B figure what can show that posterior end under the high magnification. Okay. As indicate in the beginning of this stage, it begins by a combination of two interdependent processes known as epiboli. Interdependent process, what is called as epiboli. The former involves extra embryonic spreading, and the latter the convergence and concentration of invigilating material to forms the axis, that is this one, invigilating. Okay, about the epiboli. The epiboli is the extension of blastodisc. Blastodisc. Okay, the epiboli is the extension of the blastodisc over the surface of the yolk of gastrula. Surface of the yolk of the, here the yolk, here the yolk. Right? Surface of the yolk of gastrula. Related to this process is the growth of the periblast and the formation of germ ring. Where it is periblast? Periblast. Okay. Then here is the periblast, formation of ring. Related to the process in the growth of the periblast and the formation of the germ ring. As the peripheral periblast grow around over the circumferences of the yolk, the blastoderm advances over it, but at no stage comes in contact with the yolk. Important point that, but at no stage come in contact with the yolk. The next point, that is the growth of blastoderm thus have a close relation and dependability over the periblast. On the left hand side, the same figure, figure number A, figure number B, show the early stage gastrulation under a low magnification and posterior end under the high magnification. This is the periblast, this is the rings, this is the yolk, blastoderm, investigating ectoderm, investigating endoderm. Okay, some events during the epiboly. Blastoderm cells grow over the yolk. Prospective ectoderm cells grow over and cover the yolk mass from outside, forming a layer of cells called epiblast. Called a epiblast. Then simultaneously the periblast also grow and forms an inner covering of the yolk. Here. The periblast and epiblast enclose the yolk in a yolk sac. In a yolk sac. Then about the emboli. Epiboly, emboli. Emboli is a kind of morphogenetic movement which refers to the inward migration of cells in the embryo. In this species, it is process of investigation of the rim of blastoderm. At the beginning of gastrulation, the prospective endodermal and mesodermal cells at the posterior end of the blastoderm turn inwards and migrate towards under the blastoderm so as to form hypoblast. Which form that hypoblast. However, there is a pushing of the epithelial layer and no true orientation is formed. The prospective endodermal cells laying along the posterior margin of the blastoderm migrate inward along the surface of the yolk. The last point, that is, as this cell move inward, they are concentrated towards the middle of the blastoderm. All the process is going on in the gastrulation. Okay, thank you.